Thank you. All right. Well, good morning, everyone. As most of you know, I'm Catherine Murphy, and I'm the science coordinator for the Middle Rio Grande Endangered Species Collaborative Program here in Albuquerque. I want to thank you for taking time from your busy schedules, as usual, to attend today's seminar. And we are very pleased to welcome back Dr. Rob Dudley. Rob works with American Southwest Ichthyological Researchers and the UNM's Museum of Southwestern Biology. Today, he's sharing with the collaborative program an analysis of Rio Grande silvery minnow population monitoring during 2022. And these findings are critical for silvery minnow research and for the conservation and recovery efforts of our program signatories. As always, during Rob's presentation, you might want to submit questions, and you can use the chat feature for that, or you can wait until the end and raise your hand to be recognized after the seminar. We are recording this presentation and we'll upload it later to our program portal. So with that, I'll turn things over to you, Rob.
Perfect. All right. Good morning, everyone. Thank you, Catherine and the West team. Always doing an amazing job getting these seminars going and keeping things moving along. So here we are back for another year, Rio Grande silvery minnow population monitoring. So it's a long-term project. It's been happening since 1993. And most of you probably know a lot of the background information. I'm going to speed through some of that at the beginning of the talk, and we'll get to some of the core results. But before I get started, I'd like to acknowledge my co-author, Steve Platania, also of MSB and ASIR, and Dr. Gary White, Colorado State University. So here's a beautiful photo, actually, of Rio Grande silvery minnow. It was described in the mid-1800s, part of the Pacific Railroad surveys. And there wasn't much research done over the next 100 years. Little spot surveys here and there, but it was ultimately listed as federally endangered in 1994 because of threats to its habitat and reduction of range. So the distribution, historical, found throughout the Rio Grande Basin, both in Grande and Pecos, pretty extensive range. And it was also found there with a lot of other fish listed below on the bottom right here. A total of 6 that are in this reproductive guild. And the thing that makes them unique is they all have drifting eggs, very unusual for North American freshwater fishes. And what separates them from these other taxa at the moment is that they're the only one found. So silvery minnow is the only one of these members of this guild that still remains in the middle Rio Grande, where we are. There's also an experimental population of silvery minnow down near the Big Bend region in Texas. Hasn't done too well. So what are we doing? So here we are in the middle Rio Grande. And the study area starts at the north at Cochiti Dam, ends up at Elephant Butte, and it's segmented into these different reaches, Angostura, Isleta, and Santa Acacia. At the very top is Cochiti Reach. But I won't talk too much about that today, but these are all separated by diversion dams. So just a photo. So interestingly, silvery minnow historically was found about 100 kilometers upstream of Cochiti Dam up into the Rio Chama near Abiquiu with some of the northern distribution. Cochiti changed that, so this is a large earthen dam. Fragmented the range and created a different flow regime and habitat subsequently downstream. Moving down to our first diversion dam is Angostura. It fragments the river and takes water for irrigation. And that begins the start of the Angostura Reach. Then moving down to Isleta, again, a similar story where there's water being drawn for irrigation about from March through October of every year. And also fragmenting populations. And then finally, Sanication diversion is our final diversion to the south. And then moving downstream, so this is the southern terminus of the range. So this kind of from Cochiti to Elephant Butte encompasses the critical habitat. And as you can see, another large-scale dam, one of the biggest in the world at the time it was constructed. And if you look to the left, interestingly, so that's the water coming in from the Rio Grande, the brown, slowly turning a little more green as it moves downstream, and then kind of a dirt color, dark green there. And that's just all the sediments settling out, as common for all different reservoirs, and it happens at Cochiti as well. And it changes, though, fundamentally, the composition of the water. The sediment composition is dramatically reduced downstream of these dams. And so what does that mean? Well, if you look at something like Sanication, here taking a shot in 1952 prior to Cochiti, you can see upstream of the diversion here, a broad, extensive river channel, highly dynamic, sandbed river, classic for the 1900s, really throughout the American Southwest. And then moving to the current, so this is post-Cochiti, and you can see heavy incision, channelization, loss of sediments, loss of habitats, kind of go hand in hand. And it's removed a good amount of the habitat that's most needed by this fish, which is for the larval stages. As they get into these really shallow, warm sort of habitats where they can grow up and become bigger fish. And so within this area, we do a lot of sampling, and it's more or less spatially balanced. So we have more sites in the southern reach, Sanication, than we do in the northern reach, Angostura. And this has been going on since 1993, approximately, with a couple little shuffles here and there for our core 20 sites. Moving flash forward to 2017, added 10 additional sites at the request of the program. And the request at the time was to add them so that we'd have a total of 10 per reach. So we now have adding 5 into Angostura, 4 into Isleta, 1 into Sanication, 30 sites. And we do those in April and October for more intensive monitoring. But one thing that does happen, not so much in the Angostura reach, but a little in the Isleta, and for sure in the Sanication reach, is river drying. So we have replacement sites where we can come in during any of the months that we're sampling, April through October, and find a spot. So if the spot is completely dry, we'll find a nearest neighbor that's still flowing, and sample there and see what's going on. In terms of what we do when we're in the field, we break things down into these types of mesohabitats that we're interested in. So 5 illustrated here, runs and shore runs, the higher velocity, things like backwaters, pools, and shore pools, lower velocity habitats. And then we break those down into our sampling regime and have a quota here that I've listed, more in the habitats that are more abundant, like the shore runs. And then we limit ourselves to either 2 or 4 in some of these low velocity habitats. And then break the sampling into 2 different types. So a large seine, which is targeted towards the juveniles and adults. We take 18 of those, and then a larval seine. And that's just a couple of seines, hauls. Those are a fine mesh, just looking for those, the tiniest of fish, which incidentally for all the species that we're monitoring, the Rio Grande occurs all the way from April through October, but for silvery minnow, it's really packed into June, July. Take a bunch of seine hauls at each site, standardize the mesohabitats, and pretty much do the same thing month after month. So how have things changed over time? Well, luckily, the sampling methodology, everything I've described, has pretty much stayed the same. We've had reviews over the years from the collaborative program, and then bringing in a variety of experts from around the country to kind of review not only this program, but some of the other programs as well, genetics and spawn monitoring and things like that. And the recommendations all come back pretty similar. So it's basically increase your sampling. So that was the initial sampling. We used to do quarterly, and then it went to monthly. We had another review in 2004, and the recommendations were similar, but they wanted to add 2 studies. So I'll kind of talk about those in a second to dig a little deeper into these population dynamics and to see maybe there's different ways to look at things. And then more recently, 2015, those were the additional sites, the replacement sites that I just described, and also kind of looking at dry versus not dry, things like that. Okay, so those 2 studies that I mentioned. First, population estimation. So this is pretty interesting. Occurred for these 4 years. And the idea was, well, okay, you're going out there doing seine hauls. It's fairly rapid sampling. We go out and do these 20 hauls at a site, can wrap it up with a crew of 3 in an hour. And we did pretty much the same thing with the population estimate. So we go out in October, we've got the 20 sites, we've got these habitats, similar amount of effort, but the differences were pretty notable. So there were random sites, random mesohabitats, we mapped everything, and then we did electrofishing removal sampling in the enclosures. So it required roughly about 10 times the effort, so double the crew, and it took like an entire day to do a site. But interestingly, the trends were very similar. So if you need to get population, if you need to know what the population size is, there's no substitute for this. You can't really take these EX values illustrated on the bottom for pop monitoring and turn those magically into a population size estimate that's going to be anywhere nearly as robust. But in terms of trend and in terms of understanding ecological interactions, they're quite similar. And then another study, this site occupancy study, is quite interesting. We'll talk about that later more. But essentially, what we're doing there is repeated sampling. So again, similar methods, but we go out for 4 days and sample each of the sites at the same habitats. And it gives us a more robust estimate of occurrence. So we're looking at occurrence on the top here, or as we say, occupancy. And then on the bottom, we're looking at density. But you can kind of see these patterns still hold up. We're finding a decline anyway here from 2008 to 10. Okay, so with a background, I always try to get a little bit of background just so people can wrap their heads around what the heck we're doing and how it's changed. The key objectives. So I've kind of already mentioned these, but we're looking at how are these things changing over time and space, looking at the distribution and abundance, primarily of silvery minnow. And then discharge, which is huge because it's the main thing that changes year to year and that we can measure easily, and that's large scale. So timing, magnitude, and duration of flows. And then looking at how these densities vary based on this repeated sampling. So I'll talk about that a little bit later, and also the site occupancy. And just kind of digging more into these either this kind of more detailed statistical methods to unravel some mysteries. So here we are, 93 to 2022. And we've got a nice little illustration, kind of like a clock. Silvery minnow starting here.
Natural resources.
Hi.
In February, we have a lot of adults there in the system. And then as we move into the spring, we have irrigation going, but we also have spring runoff and the floodplain inundation. We get our eggs, so that's where our egg stage kind of comes in. Then we move into June, July, that's our larval stage, and they need these sort of floodplain type habitats to really thrive. And then we move into the more uncertain season for our juveniles, where we have a combination of either monsoonal floods stable flows or intermittency, just varies across time and space. And then moving back again. So the clock starts again. And so they essentially go from egg to an adult in a year. Okay, so what happens across the course of a year in the last couple of years? You can see the green, this is historical flows, and then what actually happened in 21 and 22, below normal. And in 22, interestingly, the flows were early. So the flood pulse was not only below average, but it occurred about a month early. And that has some implications. And also you kind of see, as we move into the summer months, quite a bit below normal for summer flows. So what happened? So with our larval fish, we got what we typically get, which is a response in June, though the numbers were low across reaches and overall. And those larval fish then become juveniles, and we catch them in our larger seines by July. So typically you want to see some sort of response in July. We did not in 2022. They're there, and they're certainly there in all reaches, but they're at very low densities. And then just looking overall, so for all the different age classes, and here we have as these little red, those are our hatchery fish. You see those kind of coming in the spring, summer, and then they kind of fade out. And the aqua color laid on top now, which is in April and May, those are the fish that came in from the prior year. So 2022, particularly difficult, came in with few fish, few hatchery fish even, and then corresponding conditions. So you can imagine how it unraveled. 21, in contrast, we had a little bit better spawning response. And like I say, it wasn't stellar, but it was certainly better than 22. Okay, so kind of exciting. We've got a little bit of data that's just hot off the presses. Our July stuff just submitted to the Bureau yesterday. And so these are all preliminary data. But I want to talk about it since it is August. And it gives us the first indication. What are we talking about? So we're talking a lot about 22, but yeah, okay, whatever. But what's happening with 23 and where are we kind of in between and what are our guesses? A huge flow year, epic. It wasn't the biggest ever, but it's definitely one of the biggest that we've had, certainly in the last 10 years. And you can see that pretty obviously here with just well above normal flows during the spring. But the thing to keep in mind, and so this we'll come back to a little bit later, you can see the crash, and the crash was dramatic. So if you look at like San Marcial, talking like almost, well over 3,000 CFS, huge flows. Within 3 weeks, dry. Okay. But we had the response. Okay, so that's the important thing. So I don't want to leave you with too much, just, oh, everything's solved. It was fantastic. We had a huge response in July. I think we're going to have a rapid decline in some reaches, not all. And what we found over time is that this huge response really is very important. So in some ways, it can ameliorate some of the damage caused later on. But I did want to point that out just in terms of perhaps the water could be better managed in the summer. 3 weeks from just flooded everything to completely dry. It was pretty shocking to see. And luckily I was out there to see it. So we have these crazy distributions of numbers from zeros to just epic high. Creates tons of trouble for statistical analyses because typically you want normal distribution, not zero inflated, homogeneity or variances. We don't have any of those. So that would be the green. We have the blue. So what do? Basically break the distribution apart and analyze it as two different components because what we can do is use something called a mixture model and look at our presence absence data, which is here. So simply zeros or ones, nothing complicated about that. And it's a logistic model. So it's an occurrence model. Just were they there or were they not there? And then the second one, and this is where the zeros came in, were a problem before, we ignore the zeros. We just say, what's our density response? So anywhere where it's positive, we have a positive value, we can analyze it. And they fit our log normal distribution really well when you remove those zeros. And so that allows us to kind of dig a little deeper into these two symbols or names that we'll be kind of talking about. Delta, which is our occurrence, and that's shown as red, and mu shown as the black. That's our density. So just kind of keep those terms in mind, and you'll see they kind of co-vary, but not always. They do their own little thing here and there, and interestingly, in the past couple years, they have been going in opposite directions. So it allows us to dig a little deeper into these ecological things. What's affecting occurrence? What's affecting the abundance? And then we could take those components, because I said we basically destroyed our original data set, broke it into these two components. Well, we can put it back together and come up with a much better estimate of density. And it's not so much the density itself, because you can estimate density just by dividing the number of fish by the area that you sampled, but it's really the variance that we're most interested in. So our confidence intervals, it gets us a much more robust estimate of our confidence intervals around these values. It's a little wider than we'd normally get, and we think it's a little bit more realistic. But you see, even with these wide confidence intervals across time, that the differences across these years are so big, they essentially get washed out. So you can see there's orders of magnitudes. That's what these dotted lines are, differences across years for our density estimates. And just kind of thinking about that, at the top end of our range here, a fish every square meter. That's when they're just all over the place. When they're not all over the place, like in 22, a fish per 10,000 square meters. That's about how much we sample in a month. And just to put this in perspective, because I think this gets lost sometimes, if we were trying to measure the lowest bound, so say we wanted to go to 10 to the -3, so we're at 10 to the -2 now. What would we have to do to get there? We'd have to sample 100,000 square meters. So we go from 20 sites to 200 sites. So we're not going to do that. But it gives us an idea of how low really we were. I mean, we're catching just a few fish on these low years. And they respond in this interesting way. So it's almost like they're attached at the hip with these flows. So the flows go up, fish numbers go up, flows go down, fish numbers go down. And it's like clockwork on this scale that we're measuring, this huge scale across time and space. And at these epic changes in our densities across years, these orders of magnitude differences. And you can see from 21 to 22, the latest decline. Certainly not the lowest number that we've ever recorded, but it's pretty close. In fact, it's tied with the two of the other lowest. And the only other time we got lower was in 2013, when we had a series of really low drought years. And then, so for our parameter estimates, and you can see these are useful too, looking at things like changes across time and how those occurrence or density responses are affected by our flows. And so here, just kind of point out, so as you mentioned, 2013 was really low for our EX. 2012 and 2014, we didn't catch anything. And so that's why the red, the delta, the occurrence means silvery minnow weren't found at any of our 20 sites. We did pick them up in both of those years during our November sampling, and I'll talk about that later. That's our site occupancy model. And that's where we do 4 times the amount of effort. Okay, so bringing it back, to analyze these data and these patterns over time, we were interested then in marrying these parameters to some of the key environmental covariates. Things like this is sand less than 20 or less than 200. That's just the number of days that flows were less than 200 CFS at a certain one of our lower sites, and then ABQ greater than 3,000, how many days were flows higher than 3,000. We also have just mean flows and measures of inundation of floodplain habitats and things like that. So we have a total of 9 covariates that we add to these models. And then we mix them all together. And we also add fixed and random effects. And so we end up with about 400 models, even though they're fairly simplistic, just because we have a couple parameters, we have multiple covariates, and then we have these fixed and regular effects. And we analyze the data just using standard techniques, log likelihood and AIC. And that basically we're just assessing the fit of the data to these various models. So here's what we find. If we look at just 6 of these covariates, kind of the key ones, we end up with these interesting patterns where the occurrence probability, the delta here, very low when you have low maximum Q like we did in 2022. That's the red dot illustrated here. As the flows get higher, then we'd expect there to be them to be found at more places within their range. And then similarly, if we look at some of these other ones, days over 2,000, kind of a floodplain surrogate, and then estimated acres of inundation. And then the only ones that we find kind of this opposite relationship are these days, the low flow days. So as we get more low flow days, we find that we don't find them as many sites. Interestingly enough, so the red dot here for 22 in the bottom right here, it's a little bit off course compared to what it usually is. It just shows a little more variance in this covariate. And also, I think it kind of speaks to the fact that during 22, we had a lot of monsoonal activity. And so there weren't necessarily a series of days that had very low flows, but we had the same amount of drying during that time. And this is kind of illustrate what we'll find during these years. Like I said, kind of earlier, we even find scenes just like this in 2023, which is pretty amazing. After the floods hit the low flows. Now, this dynamic density response, though, I want to talk about. This is a little cleaner, and we get these nice relationships. Like you look at density response based on the maximum flows. It's very linear, and it goes from these low flow years with few fish and up to the high flow years. And keep in mind, these numbers are log scale since they have been transformed. And so going from, say, like a -2 to a 3, that's 5 orders of magnitude within our values. And then same sort of patterns hold for our high flows and for our low flows. They change a little bit between years, but the general patterns are similar. And just kind of an illustration of what it looks like during high flows. The water gets back into these flooded areas. So it's all like a volcano. The channel has been heavily incised. And it kind of goes up, and then there's these banks, these like almost steep sediment formed banks on the sides. And once it gets up to the top, the water actually drops out into these lower elevation areas around the river and creates, in some areas, just unbelievable amounts of flooded habitat. So it's like a threshold effect. You got to get to that stage, and then you either have nothing or you have a lot. Does the water get out of bank and get into these areas? And it's not a pond either. It's a river. It's a flowing river. You can see by the turbidity. The water's moving through here. It's a dynamic system. A floodplain is a very dynamic system. It's moving nutrients and providing just these incredible resources for fish. And in these years, it can have a tremendous response like we saw this year in the amount of larval fish that we have. So kind of digging into the details of our modeling, just keep an eye on the top 3. And it's pretty much what I've already said. There's a lot of numbers on this graph or this table, but essentially the WI, the weight on these models. So out of 400, three, which are the top three here, which all have to do with springtime flooding and our density response, they explain 90% of the overall variation. So that's key. Also key, which we kind of forget about sometimes, so you want to look at the delta response. So when it kicks in, it's not explaining as much of the variation because of the densities. But in terms of the occurrence of the fish, what really matters are the low flows. So we really start losing fish from sites over time if we have persistent low summer flows. So it's not a one component thing. You can't just produce fish and then walk away. Produce fish and then keep them to within these areas by keeping at least reasonable flows during the summer. Okay, and so what happens if we have dry sites included or not? Doesn't really make a difference. Essentially, when things are really low and bad, like they were in some of these years, removing those dry sites doesn't make a difference in terms of our interpretation of the data. They're just basically rare everywhere. They're either not there or they're in incredibly low abundance. And what happens if we add these additional sites? So this is kind of interesting. Little differences in terms of the densities estimates. So this is shown in the red. Mostly it's the error bars. So by adding more sites, you reduce your error, you get a little more confidence in terms of, how do things change across years. And so that's the primary utility that you can see between these. It's a little higher densities in the last few years for when we add in our additional sites. And I think the main reason for that is that we've lost fish in the Santa Acacia Reach, and they're a little bit heavier favored for the Albuquerque Reach. In terms of our long-term monitoring, we favor those 20 spatially balanced sites because there's no bias. And also there's such a richer data set going back to 93 to do that. Just so a couple of sidebars, we also looked at this for mesohabitats just to see, well, what happens if we broke the whole data set out and said, well, let's just look between mesohabitats and see how that shook out across years in terms of pattern. So the patterns are the same. They go up and down. There's lots in the backwaters, lots in the runs. They're just everywhere in these great years, and they're just nowhere in the poor years. And we see that here. They're always more common in places like backwaters and pools. That's a place you're likely to get them during even times when they're very rare. But the patterns are the same, and it gives us positive feedback in terms of our sampling methodology, that we are staying balanced across time and things like that. Because you can see how this could be skewed if we decided that next year all we're going to do is sample backwaters. That'd be a bad idea. In terms of long-term monitoring, we catch a lot of fish, but we wouldn't be able to match our pattern. So consistency is hallmark of science, and that's definitely true for this project as well. Mesohabitats are these models. As you would expect, mesohabitat model floats to the top with just all the weight, 9999. And then we threw in reach here. So these are two different ways to look at spatial components. So like they're either, we have a site, we're looking at mesohabitats within the site, or we're looking at a bunch of sites, and so there's reaches. So what this tells us is pretty good differences across mesohabitats, pools versus runs, not very good differences across reaches over time. And then the second way, and I'll talk about this in a second, with this repeated data. So we do that in November. We go out for 4 days, and we pretty much find the same thing. So the original, so this is 2004 recommendation from the experts. And the idea was, well, maybe, it's being a little bit biased, or there's, how much variation really picking up, and what's important in the variation, because certainly it's true across days. The habitats shift, the flows sometimes change, they go up and down. Maybe someone's a little more tired that day, et cetera, et cetera. The fish are a little moved around. We try to keep things extremely stable. So we have the same crews go out and sample these sites. We flag all the habitats, so we go back to exactly the same spots that we sampled the day before. And so what we find are that these differences are really in the negligible category, particularly when you look over time, long time, like across years, or short time across days. The patterns are very stable, and we see the same sort of dynamics over time. So that answered that question, but it'll answer another question that we'll get to in a few minutes. Okay, so we kind of throw these back into our little statistical pot, and we find that, interestingly, now our reach model take all the weight. And we did this kind of just to do a basic comparison. So you remember with the mesohabitats, didn't really do anything. We put it back in now, and it's just to kind of show that, there's reach is, there's a lot more variation that we explained there than across these sampling occasions, which basically get no weight whatsoever. Okay, so silvery minnow, we've been talking about their occurrence and their density. What about the relative abundance? So now is where the rest of the fish community comes in. And there's a lot, 40 species just about, that we've picked up in the last, I always mess the decades up on this. Joel corrected me at some point. And so we're getting close to 3 decades now. But these dynamics are really fascinating. And we find that, 40 species, just think about that. And they made up almost 80% of the catch in 2005. That's just insane. So they go from being just unbelievably ubiquitous to caves, you can't find them at all. And so we're kind of on the low end of the range currently. Things did perk up in 2017 pretty dramatically, really. And 2019 was another boost, and it's been pretty low recently. So here's a few of the other characters that are involved. Just our 10 focal taxa. We usually get about 20 typically per year, total species. So I say, we're really myopically focused on our silvery minnow here, but there's some interesting community dynamics going on as well. But in terms of the rank abundance, when we look at things, it's a pretty boring story. Look at things like red shiner, or look at things like white sucker. They're either there all the time or they're just really rare. Contrast that with silvery minnow. So that's where this coefficient of concordance comes in. Basically what it's saying is, well, if you look at all the species, they're pretty stable. These things are pretty stable over time, the ranks. And then the one exception there is silvery minnow, going from the most abundant fish in 2017 to now lower in the rankings, number 7. Okay, so final chapter, site occupancy. So this has been going on for less amount of time, 2005 to 22, which is still a long time. But like I said, this is a different study. So, we go into these habitats, like I said, we mark them with flags. And the idea is that we come back to these then habitats to the following days and do the same thing. And the fish are, put back into the habitats from where they were captured. So we can capture them the 2nd day and 3rd day and the 4th day. And why do we do this? So it's a lot of work. And the main reason really is, so if you think of those epically low years, that's kind of where this idea was born, really. We go out in October, sample 20 sites, don't catch anything. Well, okay, that's interesting. Seems like they're pretty rare, which they are. And, like I said before, you'd have to sample 200 to get to like a confidence interval of a really, order of magnitude lower density, for instance. But the interesting thing about monitoring is that when you do it, you don't catch everything. And, this is a quote from Mackenzie. I just love this quote because it speaks not to silvery minnow only, but to pretty much anything you can think of, and your spatial scale and your interest of monitoring. So if you go outside, you're like, oh, ants are everywhere in the world, and so I should find an ant. But what's your spatial scale of coverage? If it's a square inch, well, no ants. And you could go on and just like, oh, okay, there's no ants there. Same thing for things like red shiner. They're everywhere. They're the ants of the Rio Grande. They're found everywhere, most all the time, but you don't get them all the time. And we certainly get sites and multiple sites. Actually, they were pretty, believe it or not, they're pretty rare last year. So we get quite a few seine halls or quite a few sites with no red shiner. So you don't always find them, but when you do find them, you've got a data point. And if you do it 4 times, your likelihood of getting a data point is a lot higher. And so when the fish are incredibly rare, it's incredibly useful data to have. Okay, so yeah, we've kind of talked about point 2. And then in terms of the analyses, based on Mackenzie's work and then also program Mark, we used to compute these parameter estimates. So probability of occurrence, which we, in this case, we call occupancy, and then extinction and colonization. Just some incredibly important concepts in ecology. Occupancy kind of being more the more recent contribution. People were excited about this for a variety of taxa, particularly threatened and endangered species. And you can see how it plays out here for silvery minnow over time. And you see there's this progressive decline. So that's kind of the difference. The density is up and down, orders of magnitude. But as you start progressively losing them from portions of their range, that's when it gets a little more serious and it takes a little more time to unfold. And you can see what happened here across 2011 to say 2015. We had very low numbers and our occupancy levels below 0.5, which is a problem. And certainly, in 2013, down into the teens. Not as bad currently, but it's heading that direction, or was until the flows from this year. And so just kind of interesting things to think about in terms of, how does this relate to discharge? Similar sort of thing. Like I say, it takes longer to unfold, but we get the same responses where multiple years of low flows, the site occupancy levels start to decline. And then we get a high flow years, it can spike it, but it might not be instantaneous. And then certainly multiple low flow years and things start to decline again. And then if we look at our extinction colonization estimates, these are interesting. So it's basically just breaking occupancy down to its 2 fundamental components, which are extinction, or in this case, we're not targeting true extinction, but extirpation from our sampling sites. And then colonization, where they're found at those sites over time. So you can see during 12 and 13, there were they were being lost from a lot of different sites. They would just randomly show up at a new site just because we had a few fish there. But you can see these patterns kind of unfolding over time and deteriorating again in 22. Where we started losing them from more of our sites that we had collected them in 21. And you can see, so these sort of patterns, so when I talk about those 6 fish originally, those guild members, if we had this graph back in whenever they went, extirpated extinct, we don't even know for multiple taxa, but it probably unfolded much like this. Go out and catch them, it would have been low densities. And then over time, these extinction probabilities just get higher and higher. The colonization gets lower and lower. And at some point, gone. Okay, to summarize, occurrence and estimate of our occurrence and density elevated in 2019, decreases in 2020, and then further declines by 2022. So it looked pretty dire at the end of 2022. And these long-term patterns, prolonged high flows during spring, most predictive of the density, the low flows during the summer, predictive of the decrease. So you need those 2 components, the summer, the spring flows for the young fish, and then the summer flows for the juveniles. And then with our site occupancy, lost from more than 85% of its occupied sites over time. And then things declined again in 22. Okay, so what are some implications and opportunities? I always like put this up every year because I always think about it differently every year. Structural. So what can we do? Restore dynamic river flows. Well, it obviously causes structural changes. There's certain areas of the river where you can even see in 23, you just can't have flows of 10,000 in certain areas. But those are some things that are limiting the dynamic river flows of the Rio Grande. Reconnecting these fragmented reaches, that's ongoing. Reestablishing the functional floodplain, those are ongoing. So there's things that are happening right now within the Rio Grande that are positive to try to ameliorate some of these changes over time. Another one that doesn't get mentioned often, and I think about it with these dynamic flows, sediment, incredibly important in terms of shaping the channel dynamics and keeping recharged, so these areas don't get so incised. You have this clear cold water released below the dams. And like I said, it's having this progressive multi-decadal effect on the loss of habitat. These two key parameters, spring spawning, summer survival, those are what you need really to get a self-secured wild population in the Rio Grande. We're not just year to year, like, oh, wow, this is like panic, they're gone. Oh, okay, they're back, it's good. That's not the way to live. There needs to be a more sound long-term waterman strategy so that these things aren't a constant crisis as much as can be achieved. Certainly takes pressure off a little bit if you can put them somewhere else. And that's been key in the recovery plans from the beginning. Get them out of here back to where they used to be in some of these other areas. And that can help with their long-term persistence in the wild. And then finally, research. So just thinking, doing new things, all the people that are involved, we're certainly just a small cog in the wheel. There's dozens of researchers that do just amazing work, from genetics, fundamental research on, the population changes there over long terms, Fish and Wildlife Service, the Corps, the Bureau, all these different agencies, the tribes.
Hello, ISC.
Yes, and ISC, MRGCD. It's just a multi-agency collaborative that has really transformed kind of the dire situation we were in originally, and other contractors I have to mention too, SWCA, there's the different folks over time have gotten involved. And we really appreciate everyone. So our acknowledgement slide kind of captures that. Thanks, Grace. I appreciate that. Just kind of falling through the list here in terms of all the people who allow us access to the land. The contracts, they're gonna have to thank the Bureau for the core funding for the project and just keeping this project going for such a long period of time and the support they give as well. So I'll end with something I never thought I would see. I actually went out in person on July 25th of last year and took this photo looking upstream at Central Bridge. So if a picture can tell us 1,000 words, you probably could just put this up at the beginning and you would have known the story for 2022. But with that, I'll open it up to questions and yeah.
All right, thank you so much, Rob. That was great.
Sure.
All right, so Grace has her hand up.
I don't know if I'm clapping or have my hand up, and I'm sorry,
Oh,
I
cool.
didn't know I wasn't muted. So I'm going to jump in on your talk, Rob.
Hey, no problem.
As usual, yeah. I know there's some, well, first of all, I want to kind of, maybe I missed, do you have a hint of what July looks like for the silvery minnow fish? So far, our numbers looking okay for 2023, or what do you think?
Yeah, so it's good timing. So it was a little earlier in the talk. There's we just submitted it to Bureau yesterday.
Okay.
A really nice population response.
Good.
Talking orders of magnitude difference for July. So things are looking really good. We did lose, we had low flows pretty quickly after the floods. But it seems like it's been, this is kudos to the program. It's been managed in the areas where it could do the most damage. And so we have lost them from some areas in the lower area that, seem to always go dry just because of the dynamics. But some of those key upper areas are staying wet enough that we're still getting large numbers. So carrying capacity, we assume they're going to go down. They will go down by October for sure, but we're hoping things are going to look a lot better for October this year.
Okay, thanks. And then I know there's some talk about maybe increasing some of the site numbers again. Have you been talking to Reclamation about that? And what do you think that might do for the program if we do increase some site numbers? I don't know how many they're talking, but
yes, this is also very new, but I think I can touch on it and just give you the general idea. Would be there was a request to do more sites in October and April.
Okay.
And so it probably, what I would think it would end up giving us is 40 total. That's what's on the table currently. Yeah. And a little bit more spatially balanced, which we're kind of hoping for. So more in the Santa Acacia Reach, just to pick up fish down there as well.
Sounds good. Thank you.
Sure. Looks like Joel has his hand up.
Yeah, I'm not familiar with this. Anyway, hey, I noticed a lot of your mixture models test water, or they end up with results of water-related relationships between the population, either density or occurrence. Have you, since we know that you also do some sampling of eggs, introduced any egg models or egg data into that? And how does that fare as a biological factor compared to a lot of the water factors you do find?
Oh, interesting. Yeah, we haven't actually added those to our final list of models. We have added those in the past just to see how they would perform. And they didn't perform very well. Probably just because of the dynamics of, eggs versus fish. So in fact, this year is a really good example. 23, we had a huge amount of water in the river and a lot of overbanking, very few eggs. And that's a common phenomenon. I think it was accentuated this year because of the low densities of fish that we went into 23 with. They don't seem to be, the relationship is poor between those two variables. It does bring up a lot of interesting questions in terms of, the importance of these floodplain habitats during years like 23. There's even monitoring. People went out into some of the floodplain areas, couldn't even catch eggs out of those. But they're certainly there. I mean, there's no doubt there were eggs being spawned there. And I think what's going on is lower numbers of eggs, but just huge reproductive success, I think, is really what's going on. So the fewer numbers of eggs that are out there, they're much more likely to become juveniles or hatch into larvae and then hopefully become juvenile stage. So yeah, thanks.
Okay, thanks.
Thanks, Joel. Anybody else have a question? So I have one, Rob. So looking at big picture, given your familiarity with the trends for the species, on what scale should we ideally manage the silver minnow population? And I'm talking about temporal scale. Does this fish function, does the population function on a decadal scale? Does it function on a 3-year timeline? Is it literally year to year? Is it just whatever the river flows, give it's going to spawn? And that each year we get a smaller and smaller pool of genetic material. So that's a big factor. But if you were to guess or to give a good estimate of, what decadal or temporal scale these fish function on, what would you say?
Yeah, great question. I'm glad you mentioned the genetics there too, because I was about to say that that's an important thing to think about, is not just the numbers, but yeah, what are the numbers? What are behind the numbers, in terms of diversity and things like that? Because certainly going through population bottlenecks repeatedly is not a good thing for populations, but from an ecological standpoint, we're often just pretty focused on the numbers over time. And certainly having a multi-decadal sampling program gives a little bit more perspective in terms of what things can and have happened over time, and also makes you think about what things happened before the sampling program. They did make it, and sometimes our conservation ecology bristles go up when people say, oh, they made it through the 50s and 70s, but there's some truth there too, so you need to think about all these different things. Somehow they did make it through. There were multiple connected populations and different things going on back then to kind of rescue them. But they do withstand these really low years, and they come back and become really high. I mean, if I was to take a guess in terms of what needs to be, I'll give you the the least satisfying answer ever. I'd say we have to be focused on all scales. So, to really truly manage a fish like silvery minnow would be, it would be a poor decision just to say, oh, we're gonna do the 3-year approach or the 5-year approach, just because you can get yourself in a lot of trouble within just a few years. So I think there's kind of the disaster management thing going on, and so maybe that, I mean, in terms of like a rosy future where it's not ISC, or it's not the Corps, it's not the Bureau. I mean, no one has really a decision making. These things are epic. They're Supreme Court style decisions. How is the water being delivered across states and nations? By the compact. What sort of things, if there was some sort of, oh, we do actually have a multi-decadal water management toolbox. We could store some water for some of these low years and then use it. I mean, when we have the water, we could store it and then we use it in these low years. That could certainly take some of the pressure off some of these low years. What it's doing right now is we're kind of managing, the disasters as they manifest. There's certainly going to be climatic changes that we have to deal with. They're like kind of long scale. There's just drought, and it happens, and it has happened. And then there's kind of these longer term things that we're dealing with. One of the biggest threats I would say I come back to is this change in the habitat in the last well, it's been ongoing, but it's been pretty dramatic in the past 2 decades. And we see it even in years like this. We get this great flow year and all these overbank flood, and you think like, wow, this is going to be fantastic. It's going to rework the channel and all these trees are coming down, and it doesn't. It lowers the bed. And since Elephant Butte's already low to begin with, the elevations change, and we find that with some of our other studies that we've been doing with CSU and the Bureau and UNM. Those are the sort of things I think that would be the multi-decadal things to kind of try to solve, because if you can't solve the habitat riddle, you'll get these flows, but who cares? I mean, you might be able to solve, you could get a lot of habitat out of 1,500 CFS potentially. And there's been talk of that in the past, but unfortunately, the way things are right now, you have to get up to 4,000 in some of these reaches before you get anything. And it's just bad before you get to that area. It's just, it's a ditch. So anyway, kind of went off a little bit, but that's my just rambling thoughts on how to save the world.
No, that's great. That's very helpful. Thank you. I missed a couple of questions in the chat, so I'm going to go ahead and read these. Shell asks, or he says, we have a dam controlled river, yet we still have drying in a year like this because we can't hold on to water and send it out over the summer. The compact is the reason. On the implications and opportunities side, are the first two even possible given the flow restrictions forced by the compact?
Yeah, interesting question for a biologist. I don't, it's kind of goes beyond my expertise level, but yeah, certainly that's a constraint that's there and it's real. And I know other folks know a lot more about it than I do in terms of how, what are the ways to do it. I just see it as like, there's water, we got a bunch of water, it's a bank account, and then you could spend it later. It's like managing your own personal finances, is the way that I simplistically look at it, that you don't want to, come into a bunch of money, spend it all, and then be broke the next year. So if there's a way to do that, I think it would be certainly worthwhile.
All right, thanks. Okay, so Tom Turner says, great talk, Rob. The occupancy data give great insight into long-term dynamics, and the mixed model adds a lot of robustness to the analysis of distribution and abundance of RGSM. So just a comment. Grace says, this year construction at Elvado restricted storage for MRGCD, and the Corps has not allowed native water storage in Abiquiu yet, with the exception of P&P storage.
Good point.
And then Jack Marchetti says, have there been any efforts to reintroduce the RGSM above Cochiti?
No, so no efforts currently. I think there's some sampling going on. There has been sampling going on up there over time. And I think more recently, Fish and Wildlife has looked more into that question. It's a short reach, but we'll take what we can get. It's been tried in Big Bend, like I said, at some point in the talk, didn't really work out there. There's certainly the permanency of water, which is a huge plus. And it's just mostly a question of, in terms of the water clarity and the species composition and things like that. It's kind of like one of those, it's within the historic range. So it's certainly something to think about. And it's just a matter of try it and see what works. I don't know, that's mostly up to Fish and Wildlife if they want to go that direction, but it seems interesting.
All right, one last question. It's 11 o'clock. So Casey Ish says, thanks for the talk. Any plans to introduce eDNA sampling to your data set?
Yeah, well, I guess it depends what component of eDNA sampling. It's interesting on some levels. So I would say, I guess at the moment, no, but not because no, we don't believe in it or something. It's just, it's not part of the population monitoring program, or, our contractual obligations or whatever. In terms of just academic interest, I would say, yes, it's very interesting. And it's particularly interesting in terms of other taxa and at low abundances. From the genetics folks that I've talked to, it's particularly problematic for rivers in particular, in terms of trying to tease out the complexities of the kind of the meaning behind these things, because it persists in the environment for a period of time, and it's, they're drifting, things like that. It works out really great in lakes, for instance, where you want to say, hey, we're at this new lake in South America, I wonder what's here. And then you can grab some water and look at it like that. It's like a presence absence thing. Certainly would serve that purpose for the Rio Grande, I think, as well, in terms of other taxa. And the only time I've been really intrigued with it would be when we're not catching essentially anything. I could see that would be potentially its time to shine. Think disaster scenario where we, even with this really rigorous monitoring, would go out and don't catch anything. I would say, yeah, that would be the time to really emergency do that. And not to just, disaster scenario, it'd be worth probably a program thinking about it over the next, however many years, just to at least get the methods established so that it could be used down the road. I see Casey maybe has a hand up too, or that might be part of the question.
Yeah, no, thanks for the
Sure.
response, and I'll be brief. Just so you know, the district was able to partner with Audubon and World Wildlife Fund this year for the what we think is maybe the first eDNA sampling run on the Middle Rio Grande. We're
Awesome.
pending the results of those samples, but when we learn something or when we get those results back, we'll be happy to share with everybody kind of how we orchestrated the collection and what we think we learned and what we think we didn't learn. because I think there's a lot of variables out there, like Robert just alluded to. So anyway, yeah, thanks for the talk. Appreciate it.
Perfect. Thanks, Casey, for that and the follow-up. That's fantastic. Love seeing research go in new directions. And yeah, we'd love to be a part of that sharing too. Yeah, definitely send it out to the program. And yeah, it's fun to watch everything evolve over time. And that sounds like a really exciting new component.
Yeah, I'm going to go ahead and rope you in, Casey, for either presenting at our symposium in December, or when you have your results, you can provide a seminar if you want to. I'm going to put your name down. So awesome. Thank you. All right. Well, we're after time. So oh, there's some more things in the chat. I think they're just comments. So Rob, I'm going to go ahead and let you off the hook. Thank you so much for taking the time to do this today. And thanks everybody for attending.
Yeah, absolutely. Thanks again. And thank you, Catherine and Debbie and the West team. Really appreciate it. And look forward to it again in the future. So everyone have a great summer.
All right. You too. Thanks so much.